Thank you for having me. I'm Dr. Troy Lund at the University of Minnesota. I specialize in pediatric bone marrow transplant and have expertise in adrenoleukodystrophy. And so today I'm going to really talk about the goal of transplant when we perform it for cerebral adrenoleukodystrophy, and that is to get the brain engrafted. And so I'm going to tell you about one of two brand new stories in basic research coming out of my lab. So hopefully it'll be entertaining. Let's see here. So the goal, what is the goal? It's to get cells, brand new cells into the marrow, and we do that through gene therapy or bone marrow transplant. And then those cells somehow get to the brain. And in the meantime, the cells go through lots of differentiation and become and reconstitute your bone marrow. They reconstitute your blood. And then by some process, there's some cell that goes to the brain and becomes something. A lot of times we use the term microglia replacement or microglia engraftment. I'm not sure that's entirely correct anymore. And I'll show you the reasons why. All right. So what do we know about the brain and engraftment? Well, like I just said, it's thought that IBA1 positive microglia cells engraft the brain, and that's why everything works so well for neurodegenerative diseases. Though, in fact, we're actually not sure the cell type doing the engrafting or what it becomes. We also don't really know the timing of brain engraftment. Does it happen on the day of transplant, or does it happen, for example, months to years later? We also think that chemotherapy or radiation is required to accomplish this process. It certainly is for marrow engraftment, but what about the brain? So I'm going to talk about a lot about model systems, and everything I'm going to show you is in a mouse model system. The first paradigm is a bright green mouse called a GFP mouse. The bottom shows this mouse is extremely green. It glows in all tissues. This happens to be a nude version of the mouse, but any regular GFP mouse is extremely green. We often use the bone marrow cells from that mouse and place them into an irradiated wild type mouse, let the mouse engraft, and then we analyze the brain or the blood at the marrow sometime after transplant. In this case, it's 5 to 30 days, trying to get an idea about timing of brain engraftment. So we're going to keep talking about the same model over and over again with different twists. So let's just look at the blood and how that engrafts after transplant. And on the bottom, it's day 7, day 12, day 30. And from the farthest right of each of these, you can see by day 30, the blood is fully engrafted. And by day 7, it's about half engrafted. Day 7 is very early after transplant. So there's still a mixture of dying host cells and engrafting new cells. The middle graph shows granulocyte engraftment, which is very rapid, actually can occur in 7 days. And then we have a breakdown of lots of different cells on the right panel. B220 is B cells, CD4 and CD8 are types of T cells, GR1 is granulocytes. And you can see that there's a certain trend towards engraftment, which really concludes with nearly 100% engraftment by day 30. So that's interesting. This is pretty standard stuff. It's been known for decades. If we look in the bone marrow now, we can look at bone marrow cells, just the high-level bone marrow mononuclear cells, BMMC, fully engrafted by day 12, and again, day 30, partially engrafted by day 7. If we dig into the refined stem cell populations of your bone marrow called LSK, they engraft pretty robustly about a week after the transplant and then stay engrafted. So why aren't they 40% like the mononuclear cells? Because you've got a mixture of still host mononuclear cells that are dying off over time, and that dying off takes probably a week or two. So you get an admixture at day 7 before all the host cells are done at day 12 and day 30. For the stem cells, they go away pretty quickly. So again, nothing new here, nothing surprising. But what about when we look at the brain? So now we're going to look and count the number of GFP cells in half the brain. We do this by flow cytometry in the lab. So we take the brains out of the recipient animals and we mash them up, and then we run them through a machine that can give us the number of GFP cells or green cells there. You can see at 5 days, there's hardly anything. And then at 7 days, we do start to see just a few cells, maybe about 900 cells. And then it's day 12, where we see a nice number of cells, about 3,000 cells. That's about the same as day 30. And in fact, if you go out to day 90, you don't get much improvement. So what we see is a kind of a rapid upswing to about 12 days, and then it's pretty constant. And so that's, we've repeated this many times, and the trends are all pretty similar. So good numbers by day 30 with a peak, with the highest point at day 12. So that gives us some sense on timing. So cells aren't up there 5 days and not much by 7 days, but by about 2 to 4 weeks, they're up there, at least in this model. Now let's, this is a half a brain, and now this is sliced down the middle, and then we just put it under the microscope, and we can actually count the green donor cells in various regions. And here we've hand outlined the cerebellum, the cortex, the thalamus, midbrain, corpus callosum is on another section. This just, we can then analyze where the cells are going, which was our next question. And this shows you in this high mag view, I'm not making it up. The circle is our drawing, and then the little green dot in the middle, that's the engrafted donor cell. And so this is what we did, was identify all these donor cells and then counted them by hand. And this is what we get. So this is, the data is percent of total green engraftment. And so this is the distribution. Most of the cells are in the cortex at 7 days, and very few are in the corpus callosum. And again, the structures are much different in size. The corpus callosum is small, the cortex is big, everything else is in between. So it just kind of makes sense. And if you look at 7, 12, and 30 days, the trends are all about the same, remarkably, with not much difference, despite low numbers at day 7. And so then we normalize this to square millimeters. So now accounting for the smallness of the corpus callosum and the large area of the cortex, if you look at donor cells per square millimeter, you actually get pretty consistent results across the brain. And this is true at day 7, day 12, and day 30. You can see cell numbers go up quite a bit from 7 to 12, as I showed you with the whole number counts, and maybe a little bit higher at day 30, though not too much. But it does now give us a better understanding of the timing of getting cells to the brain, as well as where they are, which is pretty much everywhere. I tried to correlate marrow engraftment and brain engraftment to see if one would predict the other, and it was very difficult. I'm showing you one singular graph that was significantly, statistically significant, and that was percent of LSK or bone marrow stem cells that were GFP positive or green versus the numbers of cells in the hemisphere at day 12. And not too many times in science do you get dots that line up so cleanly on a line. Unfortunately, I don't know how to interpret it because none of the other data analysis worked out. This one was just unusual, but it could be just more or less some random type of alignment, because I just don't know the true significance of this data point and this alignment. But I thought it was interesting to show. So next, we have to ask ourselves, what is engrafting the brain? What kinds of cells? And so we can look at those GFP cells, and we can break them down into CD3 T cells, B220 B cells, granulocytes, and monocytes by these markers on the x-axis. Turns out not many of them are T cells. 10% are actually B cells. A good chunk, about a third, are granulocytes. And as previously demonstrated by many labs, a lot of them are monocytes. But it does give you some idea about the distribution of what's getting in the brain. It is clearly not all microglia cells, as I'll show you in a second. But it is a variety of cell types that are entering the brain. We can look at the same data a different way. Again, this is 30 days post-transplant. So of all the cells up there, this is now the host and the donor. What percent are donors? So if you look at all the B cells, 100% are donor. If you look at all the monocytes, about 80% are donor. If you look at all the granulocytes, about 60% are donor. There's still about 40% host granulocytes left over. And this marker, CX3CR1, is a fractalkine receptor. Fractaline's an important protein that helps guide microglia growth and other aspects of brain development. It is thought to be a marker on microglia cells. If you actually look at how many microglia cells are engrafted at day 7 using this marker, it's pretty low. It's about 1%. There's some variability, and my guess it's probably 1 to 10%, depending on the experiment. And I'll show you a more refined experiment to answer that question. So that's next. So there's a mouse model which has green protein only in microglia. And the microglia, it's an IBA1 GFP mouse. It is exclusively available in Japan. We were lucky to establish a colony a few years ago. IBA1 is a protein that's highly expressed by microglia. In this case, this is a reporter in which all the microglia are green. And the initial paper looked at developing brain and showed that indeed, yes, the green cells in the brain stained with an antibody for IBA1, which is in red, and the overlap looks very nice, which is yellow. So we got these mice, established a colony. It took about a year or two, and we did the same thing. We took a brain of this mouse and we stained the cells for red microglia, and then we looked at green fluorescence, and we found that they overlapped nicely, just like that was reported. Here is a large blowup. It's not a perfect stain because the IBA1 stain is red. And actually, if you look at microglia, they have lots of little processes, 20 to 30 to 50 different fingers come off of them, and they all stain red. So it looks like there's a lot more red staining there is, while the GFP is only in the cell body. But the overlap we thought was very good to use these mice in experiments. So our data, we thought, confirmed what was published. So now the trick is to take those mice, which are IBA GFP, they're also mismatched for another protein called CD45.2, so we can identify hematopoietic cells. We use the bone marrow from these mice, and we give it to wild type mice, which are CD45.1 in the hematopoietic cells, and they're not green. And so then we can analyze the brain 30 days after transplant and see how much microglial engraftment we got. So let's take a look at that. So what we can do again is, because of the CD45 mismatch, we can look at all the donor cells in the brain and we can count them. And we find at 1 month, there's about 1,000 donor cells in the brain. And this agreed with our prior data that we get 1,000, sometimes 2,000 donor cells in the brain, all different types. At 9 months after transplant, you actually see an upswing up to about 5,000 donor cells in the brain. But how many of those cells are true microglia, and i.e. how many are green? Turns out only about 2% at 1 month are true microglia. So these are the IBA1 GFP positive cells. And that number is actually the same when you look 9 months out. So really only a fraction of what's getting in the brain are true mature microglia, and the rest of what's being engrafted is probably a completely different cell type, maybe an unknown cell type actually, because of the uniqueness of going through bone marrow transplant with a donor. All right, let's talk a bit about conditioning. When we talk about conditioning, we're really referring to chemotherapy, busulfan, and or radiation. In the lab, we often use radiation that's equivalent to busulfan because it's a system that's been around for 3 or 4 decades, very easy to tune. All labs have irradiators, and it's very easy to customize experiments. So what we did in this experiment to teach ourselves about radiation is we took an immune deficient mouse. It has no immune system. It requires no radiation or busulfan to actually engraft. The mouse was developed to take on human cells. But we thought, why not put in some green cells? And now these mice are the ubiquitous green cells. It's not the IBA mouse. So it's just the regular stuff I showed you the first time into the mouse with and without radiation and see what happens. So we did that. We waited a month, and without radiation, these various marrow stem cell types actually show some engraftment. The high-level stem cells are at 40%. Refined stem cells, again, are at probably 5 to 10%. But again, there's no radiation involved here. This is just a syringe of cells going into the animal, and they engrafted. If you give the animal just a little bit of radiation, half the amount normal, you get full engraftment. And this is what exactly we expected to find. So again, you don't need radiation to engraft this animal, but it does help. Let's look at the brain now. Do the same thing. So this is the first data I showed you in a black 6 mouse, so a regular mouse, full 9-grade radiation. We get about 2,500 cells. In this animal, this is the immune deficient animal, 0 radiation, we actually get about 3,000 cells. So this is the first time anyone's seen brain engraftment without a drop of radiation. So cells can get to the brain without radiation. We suspect the same is true for busulfan. Those experiments will be finished in 2 weeks. Now, what happens if we just give a little radiation? A lot more engraftment happens. So what this demonstrates is that radiation busulfan is not mandatory in our model system to engraft the brain, but it certainly helps engraft the brain. It's probably playing a unique role, which has very little to do with the blood-brain barrier in my mind. In fact, has to do more with the immunology of the brain than anything else. So again, this is we're testing this all in busulfan, and we should have that data updated in about 2 weeks. This is very interesting. So the next question was, do bone marrow cells enter the brain on the day of transplant? Is that what's happening? Or could they enter the brain, or can primary bone marrow cells enter sometime after transplant? The data I've just shown you alludes to the fact that it's probably a slower process. But we did design an experiment to try to sort that out. And what we did was we irradiated, again, a CD45.1 mouse. We gave them a transplant of green cells on day 0. So that's transplant day, just like we did with all the other experiments I just showed you. But then we took another group of mice and we waited 7 days. Then we gave them a transplant. And we took another group of mice and we waited 2 weeks. Then we gave them a transplant. So again, it's whole bone marrow cells. And then a 4th group. We actually waited 4 weeks after radiation to give them their transplant. And in each group, we waited a full month after the infusion, so the cells would have the same amount of time to mature if that's what had to happen. So again, 0 to 28 after radiation, and then 28 days after infusion, we analyzed peripheral blood, marrow, and brain. So what does it look like? It looks like this. This is peripheral blood. This is marrow. These are marrow stem cell populations. These are the day of green cell infusion. So day 0, which is the standard. Then we waited a week, 2 weeks, and 4 weeks. And what you can see is that you get good engraftment even if you wait a week or 2, you actually get pretty high levels of engraftment in the peripheral blood and the marrow. In fact, if you wait a whole month after radiation, you can still get 25% engraftment in the peripheral blood and marrow. Stem cells a little bit less, but it's impressive. You can wait that long and get green cells to go there. What happens in the brain? So what happens is this is our standard paradigm to give the green cells on day 1. We get, in this case, we got 5,000 cells, But what if we waited a week to give those green cells? We actually had 83% loss of green cell engraftment. And at 2 weeks, there's actually a little blip up here. But then if we waited 4 weeks after transplant, then engrafted the mice, then waited another 4 weeks, we hardly got any cells in the brain. So there is something special about the timing. So again, despite having great engraftment at 7 and 14 days after radiation in the blood and in the bone marrow, we get almost hardly anything. We only get about a fifth of what we normally get in the brain. So again, this goes into the unique feature of what's happening to the brain after conditioning. There's a paradigm set up where there are signaling molecules, I would guess, that are calling to the cells, but the cells aren't primary bone marrow cells. They are probably a circulating mature cell type that needs to mature in the peripheral blood before it goes to the brain. Otherwise, these day 28 mice would be fully engrafted in the brain. And again, we looked at a subtype of cells here, CX3CR1 cells, similar results. We lose most of their engraftability if we wait a week after radiation. This is just a one simple experiment. The CX3CR1 can be a surrogate for microglia. In this case, we measured these residual host microglia in the brains of all these animals. So in a non-transplanted animal, you have about 125,000 microglia. If you wait 4 weeks after radiation, you drop that down to about 75,000. If you wait 5, 6, 8 weeks after irradiation, you drop it down to 25,000, but it doesn't go away in the mouse and likely not in humans. Microglia turn out to be a radio-resistant cell type. And so we can knock a lot of them off, but we don't knock them completely off. So my guess is that mice and humans are going to always have a mixture of host microglia and maybe a little bit of donor microglia. And like I explained earlier, that amount is probably around 5%, maybe a max of 10%. Let's see here. So the conclusions from this data set is that the donor cells in the brain are likely not mature microglia like we think about them or read about them in the textbook. It's a variety of cell types, and they're probably very unique cell types in the brain. Some people call them perivascular macrophages. Some people call them brain macrophages. Some people call them all sorts of different names, but they're clearly not what we think of as mature microglia. Nevertheless, the brain does engraft. The donor cells in the brain really start to appear robustly about 12 days after transplant in our model. They are distributed throughout the brain from what I can tell. The donor cells are not from fresh primary marrow cells, but actually are derived from the circulation. So the marrow probably has to engraft first, mature, release cells. Those cells get to the brain would be my best guess on how it's working. And the timing is critical. The cells probably fill a niche. They're probably following a homing signal. And the last thing I showed you was host microglia are lost over time, but there is a limit. So they're not completely replaced. And does all of this matter? If we can learn the system, can we accelerate it? Does that lead to better outcomes? Well, in fact, we already published a paper that when we looked at human patients that engrafted, those patients that engrafted the fastest actually had their gadolinium go away and faster. In this case, we are looking at gadolinium resolution by day 60 after transplant, and the patients that engrafted before 16 days after transplant had that gadolinium disappear faster. And that would be our surrogate outcome for an improved ALD remission. So the answer is yes. In terms of GAD remission, whatever that means clinically, we don't know. And then another part of this paper is that we looked at donor chimerism. So 70% donor cells have the faster resolution of gadolinium. If you're only 50% donor cells, you are less likely to resolve your gadolinium. And again, we use that as a surrogate for ALD remission. So the implications of all of this for ALD is that a small amount of donor cells contribute to disease stabilization is what we believe. And some of this data can probably lead to further development of that idea. Conditioning plays a novel role, not what we thought it did. It really changes the immune system of the brain per se. Does accelerating engraftment improve outcomes? Well, if it did, maybe we could do a better job on more advanced CLD patients. And maybe this is applicable to gene therapy. And why do I ask that question? Because only a minority of the cells are corrected with gene therapy, and perhaps getting more of them into the brain might even be better than what we're doing today. And that's all I have. Thanks.